Okay, we're going to start out with the second part of our midterm post-exam review, and we're going to move on to the next question. So during protein synthesis, the peptide chain is synthesized in the, and if you look at these answers here, you should immediately be able to cross off the 3' to 5' direction, because 3' to 5' is specifically a nucleic acid term. Now the next one up, phosphoryl to hydroxy direction, that actually can be crossed out too. That was the distractor answer. Sounds a little bit like it might be the right answer, but it's actually not. So you really should have been able to rule these answers out to these two answers, the amino to carboxyl or carboxyl to amino, because you're talking about proteins here. And proteins are the N or amino to C or carboxy termini. So this was actually the correct answer for this question. Now the next question, which of the following is more likely to result in a phenotypic change? A frame shift mutation at the beginning of a coding sequence, a frame shift mutation at the end of a coding sequence, a missense mutation at the end of the coding sequence, or a silent mutation in the middle of the coding sequence. So hopefully the one you could cross out right away is a silent mutation in the middle of the coding sequence. And the reason is a silent mutation is a mutation that doesn't change the nucleic acid. So if you have a bunch of, it doesn't change the amino acid. If you have a protein, string of amino acids, and this triplet codon here has a silent mutation, it's not going to change this amino acid. So it's likely not going to generate a phenotypic change. Now the other thing you want to look at is mutations at the end or the beginning of the coding sequence. So mutations at the end of the coding sequence, unless it's a silent mutation where you're not going to change nucleic acid, other mutations at the end of the coding sequence are going to be less likely to generate a phenotypic change than those at the beginning. So a missense mutation at the end maybe could generate a phenotypic change, but it's asking which of these answers is more likely to result in a phenotypic change. So I would narrow it down to a frame shift mutation at the beginning or a frame shift mutation at the end. So remember, you have your ribosomes that are coming down and translating your RNA into protein. So let's say we have our triplet codons GCC, ATC, CCA, GGC, CGC. So you're reading or the ribosomes are reading and reading these. Each one is an amino acid. Now let's say you have a frame shift mutation, and in the frame shift mutation you actually, for this frame shift, you're actually generating a nucleotide here. So what's going to happen is your triplet codons are going to shift. That's what a frame shift is. It's shifting the frame, the reading frame of your triplet codons in translation. So instead of there being the GCC, which you would look at your amino acid chart and figure out what these new amino acids are, now it's going to be translated as GTC, CAT, CCG. So when you have this type of frame shift mutation at the beginning of a coding sequence, from the frame shift all the way down, you can get completely different amino acids. So this answer, the frame shift at the beginning, is going to be the most likely type of mutation to generate a phenotypic change out of these answers. So the next question was, the original DNA is A, G, A, C, G, C, C, G, A, G, G, A, and during replication it gets mutated. What type of mutation is this? Now again, for this question, before I went and looked at the answers, I would really compare these sequences to see what's going on here. So I would write them out. A, G, A, C, G, C, C, G, A, G, G, A. And our other mutated sequence is A. Okay, that's the same. G, A. Oh, now we have something different here. We have G, C, and then G, A, G, G, A. So in this situation, we're missing this C, and we are missing this C here. So this example is an example of a deletion mutation because there are deleted nucleotides. Now in inversion, we would see this whole section may have been flipped or inverted, and we didn't see that. And insertion would have been if we had a new nucleotide present. So let's say in this area we had a T, and in this area we had an extra G. That would be an insertion, and that's not what happened. And then a point mutation would be just one out of the whole string of nucleotides was mutated. So let's say this nucleotide was actually a T. This would have been an example of a point mutation. So what this was, and you really had to draw this out and compare the two sequences, was a deletion mutation. Okay, the next question, using the genetic code and the rules for translation, what amino acid would be made from this sequence here? So now there were two clues in this question. One, in big capital letters, the rules for translation, and then another direction here. Be sure to follow the rules. So when I read a question like that, I'm thinking, okay, there's something specific that's being asked here. Make sure you follow the rules. So we're going to go to the next slide for a moment because the sequence is actually here right at the top. This is the same sequence that was in the question, and here is a genetic code. So now when I put in there, make sure you follow the rules. Remember, you have your messenger RNA, and you have your large unit, small subunit of your ribosome moving down your messenger RNA and translating your triplet codons. So a ribosome is going to be coming down, and a ribosome, here's the rule, is not going to start translating until it gets to a start codon. It needs a start codon to begin translating. So some of you started to translate this immediately. So G, here's first, C, which is right here, is alanine. Well, some of you actually said that was the answer, the answer that started with alanine, which is not correct. You had to wait and move to your start sequence, which is your A, T, or U, G, which is methionine or a start. So we have methionine as our first amino acid. The next triplet codon, GGC. So we come over here, GGC, I see it right here, is a glycine. The next one, GTG, we go GU, G, it's a valine. We have ATT is our next one. So AUU is an isoleucine. Now GCC, we have GC and C here. I see it here. GCC again, we have another alanine. GGG, which is right here, GGG, it's a glycine. And TGA. So now here's the other rule. T, G, and A is right here, and that is a stop codon. So you should have read this sequence starting right here at the ATG, and you needed to stop reading it right here because this is a stop codon. So your sequence was methionine, glycine, valine, isoleucine, alanine, and glycine. So let's go back up to the answers. We have now our answers, and some of you got tricked with the alanine first, that GCC, and that was not the right answer because you had to wait for the methionine at the ATG. So for those of you that were following the rules, you should have immediately been able to cross out this one and cross this one out because they didn't start with a methionine, and a methionine is a start codon. So you should have been narrowing it down to 2, and that's always the case with some of these multiple choice questions. Personally, I always have had difficulty with multiple choice because you can always narrow it down to 2, and then those last 2 can be very tricky to get the right answer. So we have methionine, glycine, valine, isoleucine, alanine, and glycine, and this one was the correct answer for this question. Our last question that we're going to go over, a molecular biologist needs to separate pieces of DNA that are between 50 and 150 base pairs in length. So that's the key part of this question. Which method should be used? Okay, so you're looking at the answers here. The one thing you'd immediately want to decipher when you're thinking about a size of a nucleic acid and what to run on. So the first thing is what type of product are you running? And it's DNA. So when you see you're running DNA, there are 2 things I can immediately cross out here. One is 2-dimensional gel electrophoresis, and one is gradient denaturing electrophoresis. And the reason is these 2 are for proteins. You don't use these for DNA. So you should have been able to cross out those 2, and you're left with polyacrylamide and agarose gel electrophoresis. Both of these can be used for DNA, to run DNA on a gel. Now, the thing about the 50 to 150 base pairs in length, that is a very, very small piece of DNA, a very small fragment you're running. So if you've been listening to the voiceover lectures, you would have remembered we discussed very small fragments need to be run or are better separated using polyacrylamide gel electrophoresis.